Hello and welcome to the third webinar of our Let's Talk Disease series, presented by the Ontario Ministry of Agriculture, Food and Rural Affairs and Greenhouse Canada magazine. Now, before we begin, take a look at the GoToWebinar control panel on your right. Click on the little arrow on the Questions tab near the bottom. This is where you can submit questions throughout the webinar. This is also where you can respond to the presenter, as she will be asking you questions throughout the presentation. A recording of this webinar will also be available on our website at greenhousecanada.com and sent out after 24 hours to anyone who's registered. Our webinar today will be moderated by Dr. Sarah Jandricic, who is the Greenhouse Floriculture IPM Specialist in Ontario, responsible for both IPM of insects and plant diseases. Welcome, Sarah.
Hi, everyone. I'm really excited to have Dr. Rane with us here today to talk about disease diagnostics in ornamentals. She's going to be showing you lots of pictures of sick plants that you can enjoy, but hopefully you don't have in your greenhouse right now. Karen is currently the director of the University of Maryland Plant Diagnostic Laboratory, and she has more of 20 years of experience in plant problem diagnosis, specializing in diseases of ornamentals. She has authored numerous newsletter articles and conducted workshops and presentations on disease diagnosis and management for growers and green industry professionals. Take it away, Karen.
Thank you, Sarah. I'd like to say from the outset that you may need to raise the volume a little bit. We're trying a phone issue here, and I hope everyone can hear me loud enough. But thank you for the introduction. And as Sarah said, I'm a plant diagnostician, which is sort of like being a plant detective, actually. But I've been doing this for actually 20 years is an underestimate, probably more than 30 at this point in time. And most people don't want to have to deal or have to actually contact somebody like me. One always hopes that everybody's crop comes out fine, perfectly, with no problems. But as we all know, occasionally you run into issues. And the point of this talk is to try and help with determining what the causal factors might be with a particular problem. Now, we've got different groups of causal agents that can cause problems in greenhouse crops. The two factors that are biotic or living organisms are the insects and mites, all the arthropods that can cause problems, and the plant pathogens. Those are the microbes that cause plant disease. But there are other factors that cause disorders in plants as well, and when you're diagnosing a problem, you have to take all of these things into account. There are issues with perhaps environmental stress factors. There are also problems with chemicals, whether it be pesticides, issues, or sometimes things from outdoors that come into a greenhouse that may be pesticide related as well, and nutritional problems. So those three categories, the chemical, the nutritional, and the environmental, are the causal factors that are non-living or abiotic. And you can imagine within each one of these categories, there are numerous specific individual problems within those categories. So trying to discern what is actually going on can be a really daunting task. You're trying to sort through a myriad of potential causal agents. The process of figuring that stuff out is called diagnosis. And I like the definition of the art and science of identifying plant problems using symptoms, signs, and case history information. The science part is the part that you learn as growers over time, your experience with the crop and what your school or education may be and what references you have. And the art part just comes with experience. There's no substitute for learning on the job and documenting the different things that happen, and so that you can refer back to those things and as a learning process. Why is an accurate diagnosis important? Basically, it's to know your enemy. It's really important to know what the problem is in order to make effective management decisions. Some reasons for that pesticides won't fix problems caused by those non-infectious or abiotic factors. Pathogens differ in host range, so it's important to know what you have, to be aware of whether it can spread to additional crops in your greenhouse or not. Diseases can be spread in different ways. Different pathogens have different pathways to gain access into plants. And so again, cultural practices, the choice that you make for those cultural practices depends on the diagnosis that you have for a particular problem. And finally, any kind of pesticides you may use, fungicides or insecticides, they're selective. There is not one single magic bullet to care for all of the insects and disease issues that you might have in a greenhouse crop. So that's why identifying the particular causal agent is so important. I've distilled the process of diagnosis down to 4 steps. And those of you that have a lot of experience in growing plants probably do this subconsciously, but I thought it doesn't hurt to go back and kind of reevaluate how you solve a problem or how you try and discern what the causal agent is. And this stepwise process I find to be fairly useful. And the first step is know the host plant. That sounds kind of obvious, but it really, if you know the host plant, it can really help you to narrow down specific problems. First of all, when you know the host plant, you have to know what's normal. And pathologists like myself just struggle over some of these cultivars that are variegated or have different growth patterns, because one could look at those and think that they're symptomatic. But if you know this is the cultivar that you chose, you know that this is normal. The poinsettia picture, for example, the picture on the right, has some curling leaves on the one cultivar and it has variegated leaves on the other. And both of those are normal for those particular cultivars. But someone who didn't know that might think there was some sort of a problem. Similar with the heuchera on the left, that's quite a strong variegation. It could be mistaken for virus problems, but really this is normal for the plant. So when you know what's normal, you know, and you see symptoms like this or what look like symptoms, then you can rest assured that this is the proper growth for that particular cultivar when you know what the plant is supposed to look like. Secondarily, when you know what the host is, you can use that to get a list of the common problems that occur on that host. For example, if you're growing pelargoniums or geraniums, it's quite common knowledge that geraniums are very susceptible to botrytis blight, which is the picture on the left there, and also to edema, which is the picture on the right, which is a non-infectious disorder. But if you know the host plant, you know the common problems that you might anticipate, and that goes a long way to narrowing down that long laundry list of issues that you might start out with in trying to diagnose the cause of a particular plant symptom. The second step in diagnosing plant disorders and diseases is to check for symptoms and signs. Now, a symptom is a plant response to an agent. It can be a response to a biotic agent like a disease microorganism, disease-causing microorganism, or it could be a response to adverse environmental or cultural conditions. A sign is the direct physical evidence of a pest or pathogen, things like cast skins or fruiting bodies or things like that. That's what we call a sign. Symptoms is kind of the language of plant pathology. And we're going to go through some symptoms, some examples of symptoms here that so that we're all on the same page when we use some of this terminology. It helps when we communicate to specialists what the disorder, what you're seeing. It helps when you know what these symptoms actually mean. So we've got 2 plug trays here, 2 flats that are showing the symptoms of damping off, which is a very common problem on seedlings. It can be a collapse of the young, small seedling stem at the soil line. Sometimes it's pre-emergent damping off, and that means that the seedling is killed before it even emerges from the soil. You see them either shrivel or collapse and fall over, and this is what we mean by damping off. Leaf spots, another very common symptom. And leaf spots can be somewhat difficult to diagnose. There's many different causal agents that can cause dead or necrotic areas on leaves. Sometimes the leaf spots are tan in color, like the picture in the upper left there. Here we go, right here, which is spray injury on petunia leaves. Sometimes they're dark brown, as in the septoria leaf spot, which is a fungal disease on veronica. And sometimes they have yellow halos, which is a bacterial leaf spot on zinnia here in this picture. Not the type of leaf spot can give you some clues, but there are some lookalikes, so you do have to be careful. Not every bacterial leaf spot, for example, will develop a yellow halo. Some fungal leaf spots will develop a yellow halo as well. So these are important to note, but not necessarily specifically diagnostic, but it can point you in the right direction to a particular causal agent. We also have a symptom called marginal necrosis, which is necrosis means dead. So we've got brown tissue around the leaf margin, and that's what we mean by marginal necrosis. Again, sometimes these symptoms are caused by diseases, infectious diseases, or sometimes even insects. Something that affects the stem of the plant can interfere with water transport and can give you browning of the leaf margins. And sometimes temperature fluctuations, cold damage, for example, or accumulation of fertilizer salts can also give you this kind of a symptom. But the two pictures here in this slide show you what we mean by marginal necrosis. Now, some symptoms are quite diagnostic. And the symptoms on this page here really indicate very strongly virus problems. There aren't many other factors that can cause these types of symptoms. Leaf mosaic or mottle up here in the upper left, this uneven green coloration. Mottle is pretty self-explanatory, but the term mosaic means areas of light and darker green. Here we've got ring spots, and they can be necrotic, as the impatiens here that we're seeing. And often, very often, there's green tissue in the center of the area. That's what makes it a ring spot. There's necrotic or in the case of the epimedium here, chlorotic areas with dark green tissue in the center, and that's a ring spot. And to be honest, I don't know of any other causal agent off the top of my head that causes these kinds of symptoms besides viruses. It may not be indicative of a specific virus. Many viruses can cause ring spot symptoms. But if you see these symptoms in your crop, you should start thinking along the lines of a virus issue. Similarly, with these line patterns that are yellowing or chlorosis here on this is a peony leaf, and it happens to be tobacco rattle virus in this case. But the pattern of sort of wavy lines in groups like this are very distinctive and very suggestive of virus problems. Some more symptoms to be familiar with: chlorosis. Chlorosis means yellowing. Sometimes it's an individual stem, like it is with this petunia. Sometimes it's over the entire plant. But when we say chlorosis, we mean a yellow or a very pale greenish yellow color to the foliage. Wilting, sort of, again, self-explanatory. Flaccid leaves, collapsing leaves, soft to the touch and often off color, kind of grayish. That's what we mean by wilt. It's not strictly a downward curling, because you can have downward curling, but the leaves stay firm, and that's a different kind of a symptom. With wilt, the leaves are flaccid. They clearly are under water stress. And the central picture, the lower central part, is another symptom that we call stem canker. A canker is a dead area on a stem. It doesn't necessarily suggest a specific causal agent, but this is a symptom of stem canker. And you can see the stem is green here. There's a definite margin between the brown discolored area and the healthy green tissue, and that's characteristic of stem cankers. Some other unusual symptoms that you may not see very often, but are kind of descriptive or distinctive here. we've got two different kinds of gall. The picture in the upper left is also called fasciation or leafy gall, and it's basically a proliferation of shoots. There's a swelling, but it contains lots of partially developed growing points. This happens to be leafy gall caused by the bacterium rhodococcus. Below that, on the lower left, is a different kind of a gall, and that is a smoother tissue. The tissue is undifferentiated in that gall at the base of this leucanthemum plant, and that is caused by the bacterium called Agrobacterium synthethaciens. This is crown gall, and there isn't any leafy development on this gall. It's kind of smooth. It's like an excess of callus tissue, sort of, in that lower part of the plant. And then we can also have the symptoms called distortion. And that's the picture here of the New Guinea impatiens. And the leaves are strap-like, abnormally elongated. The leaf margins are curled under, again, abnormally. And they have an unusual, the youngest leaves have an unusual shape. They're not in a normal shape. This happens to be broad mite damage on New Guinea impatiens, but certain responses to growth regulators and pesticides can sometimes do similar symptoms. Stem rot, crown rot, very, again, self-explanatory. Rot is necrosis, and you can have collapse of stems throughout the entire length of the stem, like in the sedum. You can have collapse at the base. That's usually what we mean by crown rot, discoloration. And sometimes it stays firm, and sometimes it's soft and mushy in those tissues. And root rot, again, discoloration, brown roots. These symptoms aren't specific to a particular causal agent. You might think along the lines of infectious disease if you see these symptoms, but it's very difficult to look at a particular plant and say, oh yes, that is caused by such and such a pathogen when you see these kinds of discolored areas. And the same with root rot, discolorations. There's a group of pathogens that can cause problems with roots, but usually when you're just looking at them, you notice that the color is off. You see dark lesions here. Sometimes the roots will appear white, but actually the cortex tissue has rotted off, and all that's left is a vascular cylinder. And even though it's white in color, the root is non-functioning. And that's what we sometimes call rat tailing, where that you can see that it's only the vascular cylinder, it's a very thin thread on the root that's exposed, and the rest of the cortex has disappeared. And as I said, these aren't specific symptoms to a particular causal agent, but they can lead you in certain directions to look for the root rot pathogens, or maybe check nutritional fertilizer issues, because high salts can burn roots as well. Now, there are symptoms that can be distinctive to particular feeding injury from arthropods. The symptom called stippling is this little sort of flecking, light-colored small spots across the leaf, and that is very often caused by the feeding activity of spider mites. That's the picture here on the left. You can also have chewing damage down below here. You can actually see the little critter that is responsible in that case. But the symptom is a loss of the cortex on the lower stem, and there's also some sort of chewed tissue, some debris in there. And then you can also get leaf mining with insects, which when these particular insects feed between the two layers of epidermal tissue on a leaf, and you get that kind of circuitous striping in the leaf. So you have to remember that you're not only looking at disease issues when you're doing diagnosis. You've got to keep all the different causal factors in mind, and that's why I'm showing you some symptoms of insect pests as well. Now, those were symptoms. Those are the plant's response to a causal agent. The signs are the actual physical evidence of a causal agent. And with arthropods, insects and mites, it just may take a hand lens to tell the actual causal agent, things like white flies and fungus gnats. That was what was responsible for the chewing damage in the previous slide. You can see the actual insect and you know what the problem is. Sometimes you're just seeing a part of the life cycle, like with mice, you're seeing the eggs, or sometimes you're just seeing the excrement. In the case of thrips here in the lower right, these little black dots are the excrement of thrips, and that sort of scarring, this little light colored tissue here, that is the scarring type feeding injury caused by thrips. With disease organisms, particularly fungi, these are the kinds of signs that we would see. You can see mycelium or hyphae growing over the surface of plants, such as in this picture here. That happens to be Rhizoctonia, and we call it web blight because it kind of looks like webbing when it grows up over the surface of the plant. The picture in the upper right is a sort of a fluffy white mycelium, and you may be familiar with this disease. This is downy mildew on garden patients, and that is sporulation of the causal agent, causal oomycete, which is a fungus-like organism, on the underside of the leaf. And you can see that with the unaided eye or a hand lens. And we also have white mycelial growth for powdery mildew, usually on the upper leaf surface, but can be on both surfaces. And it looks a little different. It's maybe a little flatter than downy mildew. But this is the actual causal agent. And when you see these kinds of signs, you know the kind of pathogen that you're dealing with. Here are some more. The picture on the left, a very, very common fungus. Everybody should know what this is. This is botrytis, and the sporulation of botrytis. It kind of has that, also called gray mold for obvious reasons. Sporulation looks kind of gray and gets quite fuzzy. We also have some resting structures for fungi, and that's the picture in the upper right. Those are sclerotia. The little sort of red-brown round structures are the sclerotia, or the resistant resting structures of the fungus called Sclerotium welsii. And those are very persistent, and they survive for sometimes years, and that can be the source of disease problems later on if those aren't removed. And then when you look at an individual leaf spot, sometimes you'll see little black dots in the center of it. And those are invariably spore structures, sometimes pycnidia, sometimes others, of a causal fungus. And with a hand lens, those can be easily seen. So those are the signs of pathogens that we might see looking at a sick plant. So we know we've looked at a plant, we know it's not normal, we've checked for the symptoms and the signs. Now you're going to check for patterns, and patterns are really important in helping with diagnosis. In general, we're talking two different kinds of patterns, random and uniform. And we're looking for patterns of symptom development in various stages. We're looking at the entire crop or whole greenhouse bench, sort of the big picture, all the way down to an individual leaf, an individual plant part. Patterns can provide important clues. In general, with infectious diseases, there's a random distribution of symptoms in a crop or even on a plant part. Often diseases will affect only one species of plant, sometimes even just a cultivar or two of a particular species. And not all the plants will show symptoms at the same time. There will be spread over time. This is different than most non-infectious disorders like nutritional problems or environmental stress. In those cases, the symptoms are more uniform in distribution in the greenhouse or through a crop. Often more than one species of plant can be affected. If you can think about something like a temperature problem, if there's a heater failure in a greenhouse, anything would be affected by that. And oftentimes, all the plants in an area may show injury. And it can show up within a few days of the actual episode of stress, either weather or cultural practices. So in general, infectious disease is more random in distribution, and non-infectious disorders tend to be more uniform. And here's a couple of examples. We've got poinsettia scab here, random distribution on a leaf, and that's a fungal disease, as opposed to the marigold picture, which is sort of a bronzing, and that's an iron or manganese toxicity, a pH-related problem, so an abiotic factor. It's affecting the leaf as a whole compared to the scattered spotting with the fungal disease. We also get random versus uniform across the crop. In the case of bacterial blight of geraniums, we've got individual plants that are collapsing and showing symptoms while there are healthy plants next to it. But with a nutritional disorder, as you can understand, that they're all the plants on the bench are receiving the same fertilizer, they're all going to show the same nutritional disorder if they're in the same species, if it's the same crop. So that's a more uniform distribution of symptoms. Here again, we've got seedlings and we've got damping off, and there seems to be areas. There's a section here, a section here, here, and here, more random in distribution across flats. With the case of the melon seedlings here that are on the right, sometimes you may not even know if you didn't have them to compare that there was a problem. But there is a growing medium issue. It's fertilizer in the growing medium that wasn't evenly distributed. And so certain plants got a bigger dose of fertilizer than others in certain flats. And you can see the difference in the growth stage and the size of the seedlings in these plants. And that's more uniform and therefore a more reflective of a non-infectious problem. There are exceptions to every rule. That's what makes it a good rule, I guess. And that's the fact with root and crown diseases. You can get uniform symptoms. The whole plant could show symptoms, but really the problem is biotic or infectious disease as opposed to an abiotic problem. It's just that whenever there's an issue with the roots, it affects the entire above ground portion of the plant. And so the symptoms may be uniform, but it's still still an infectious disease issue. Now it's time for you to chime in, and I've got a question for you here. And if you can go to the question box and just type, what do you think? Is this a random pattern in the flat, uniform flat pattern, or no pattern? And if you can just type A, B, or C into the question and section, and we'll see what everybody seems to think. Okay, we've got A's, excellent. I'm seeing quite a few A's. It seems to be what people are going for, and I would say, yeah, that is it. I would agree that that's random. We've got healthy plants and sick plants here in the same flat. And I would like, if you dug out some of those more stunted kind of collapsing plants, this is what you would see. And the roots are looking on the dark side. Wouldn't you agree? They're kind of stunted on the plants that are stunted, and the roots are blackened. And if you took those to the microscope, you would see this. And these are the characteristic spores of Coreopsis, which is a fungus that causes the disease called black root rot. If you know you're dealing with pansies are particularly susceptible to this disease. So again, know your host. That narrows down the possibilities. Blackened roots suggest potential of this disease, especially if you see indiscrete lesions. And then you can, if you've got access to a microscope or send it to a diagnostic lab, you can have that confirmed by the characteristic spores that you see in the roots. They kind of almost look like a roll of candy, dark spores. And there's not, pathologists love this disease because there's not too many spores that look like that. Easy to diagnose. Okay, we've got a second one here. And I'd like you to see, is this an infectious disease or a non-infectious problem? And this is on Coreopsis. So type in A or B. What do you think? This is a little more tricky because we do, I look at the pattern on the leaf, on the upper leaf surface, and it's looking kind of random, scattered. But let's see, we've got sort of evenly split right now between infectious disease and non-infectious problems. I've got some comments about disease because it's dark. The spots are dark. Sometimes color can be a little deceiving. This is actually a non-infectious problem, and one of the ways you can tell is that I also have a picture of the lower leaf surface here. This is spray injury. It's only on the upper surface of the leaf. Most leaf spot diseases will actually infect the tissue, and you will see some sort of discoloration on the underside of the leaf. But in the case of spray injury, many times it doesn't penetrate all the way through the leaf. And so you have discoloration on the upper surface and nothing on the lower surface. So a little bit tricky because the pattern of spotting was more indicative of infectious disease, but the fact that the undersides were clean leans towards non-infectious problems. Okay, so we've done the first three. The final step in doing a diagnosis is to review cultural practices. And this really involves thinking about the timing of symptom development in relation to some of the things that have been going on in the greenhouse. For example, environmental conditions. Was it particularly cold and cloudy for several days before you started seeing some gray, fuzzy mold on your geraniums, for example? Or was there is there a problem with irrigation? Did things get skipped? Timing was off or something like that, and then you started to see symptoms in the plant. How about your fertilizers? Was there a problem with the injector? Maybe things didn't get fertilizer like they were supposed to. Things to think about that can indicate some of the abiotic or biotic issues that might be contributing to the symptoms that you're seeing. Some other factors to consider are, did the symptoms occur after some sort of an application, a plant growth regulator, a pesticide, even a biocontrol? There are some cultivars that may be sensitive to applications, even to like the inert ingredients in a particular formulation of a product. And so if you see a lot of symptoms a day or two after an application, that might suggest you might be dealing with a phytotoxicity issue. Pest monitoring is incredibly important. If you see a spike or an increase in your thrip populations in your greenhouse about a week before you start seeing strange ring spots on your impatiens or dark lesions on stems, that might be indicative of one of the tospoviruses that are spread by thrips. So again, important factors to consider when you're trying to do a diagnosis. And also, not just the activities within the greenhouse, but the activities around the outside of the greenhouse as well. Sometimes growers can, they're doing something in the roadway, or they're doing stuff to the plants that are outdoors that may drift into a greenhouse, and the crop inside may be more sensitive. So it's important to think about the cultural practices that are happening all around when you're trying to figure out what's happening. A good thing to do is to check for some potential problem areas in the greenhouse when you start seeing symptoms in your crop. Look for weeds, because we know that weeds can harbor insect pests as well as some of the plant pathogens. Are there dirty areas in the greenhouse? Leaving plant debris behind, again, encourages pests to continue from crop to crop, pests and diseases as well. And if you look really carefully, in some of the weeds, this might be the fellow that you see, which could indicate why you're having a problem on the crop, because there are these things underneath. The thrips are, basically carrying from crop to crop on weeds and able to provide, cause damage in subsequent crops as well as transmit viruses. Useful tools to have for plant diagnosis. These are things that help. Obviously, hand lenses and digital microscopes are great. You can even use your cell phones, and there are microscopes that fit onto the camera lens of the cell phone that do a nice job as well. Anything that helps enlarge, because most of these pests and pathogens that we're looking for are, produce structures that are quite small. So those are really invariable, and anybody should have a hand lens on them as they're walking through their house, their greenhouses. We like to use a digital microscope that attaches to a laptop, and you can get a bigger picture that way, and you can take pictures, and you can use them for training for your workers as well. If you have some pictures, help them to see what they should be looking for. So there's quite a number of those out on the marketplace. Everyone should have pH and EC meters. You need to monitor through different methods the nutrient status of the crop. It helps with to make sure that your fertilizer is being applied properly and that the plants are under optimal nutrition. References are extremely important, and there's lots of different kinds of references that are helpful. The pictures here are for the APS, the American Phytopathological Society Compendia Diseases, but there are many textbooks, and there are many publications from Extension and other grower organizations and things that can really be useful. Monitoring for pests, as I said before, is critical for helping in getting a big picture of diagnostic issues in your particular greenhouse. Yellow sticky cards, blue sticky cards, other ways of monitoring. And for those that regularly grow crops that have particular problems, or growers that have a history of a particular problem, sometimes these virus test kits, these serological test kits, are very useful. They're grower friendly. They're not very high tech, but they can help you identify particular viruses very quickly and help you manage those problems quickly. So you've done all of this and you've used your references, but you're still not quite sure what the problem might be. What is the next step? It's to contact a laboratory. And in Canada, two that I found online are the Laboratory Services Division at the University of Guelph, and also the BC Ministry of Agriculture has a plant diagnostic lab as well. And I've got their contact information here, but you can Google them and find them online. And if you happen to be a United States listener, you can go to the National Plant Diagnostic Network website and find the state lab that cooperates as part of the National Plant Diagnostic Network. That's the lab I run in Maryland as part of that network. And you can find someone to send the plants to. Now, when you're using a diagnostic laboratory, it's really important, that the lab is dealing with a sample. And so that sample has to be representative of the problem. It has to show the symptoms that you're concerned about. It has to be in good condition. So that means prompt shipment. And it has to be accompanied by the information, what you've done to that particular crop. The diagnostician is going to need to know that. If there have been pesticide applications shortly before a sample was submitted, that could have an impact on the ability of a lab to detect certain pathogens. More is always better when it comes to diagnostic samples, whole plants, plug trays showing a range of the symptoms. So the picture of the English ivy plugs on the lower left here, that's a really good sample. What is not a useful sample is a dead plant. Too many secondary microorganisms can invade plants that have been dead for some time, and that really camouflages or masks any particular causal agent, an aggressive pathogen that might be there. So you could easily miss a pathogen on a plant that's been dead for quite a while. Packaging is important to make sure the plant arrives in good condition. Using a plastic bag to keep the soil at around the roots, but not into the foliage, that's helpful. Wrapping the whole thing in dry newspaper, packing in a crush-proof box, and using a shipping medium that minimizes transit time. That's always the best. Avoid shipping over a weekend, and try not to use snail mail where things will take several days, especially in cold weather or extreme hot weather, because the packages become vulnerable to those temperature extremes and the plants will degrade. Most diagnostic laboratories have submission forms that they want you to fill out, which includes the information that we've been talking about, that case history information, and also the patterns. Is this a random problem? Is it only one crop, one type of plant? Is it multiple plants? When did the symptoms first occur? That information is very helpful for the diagnostician in the lab, as well as for you diagnosing in the greenhouse. And when it gets to a lab, these are the kind of tests that happen, that are done. We use microscopes, higher powered than what can be done with handheld digital microscopes for the most part. And so we look for particular spore structures or bacterial streaming in the case of bacterial diseases. We also do isolation tests if necessary to use several selective media to pull out particular pathogens. Serological tests, and similar to those little dipstick tests that you can use in the greenhouse itself, we'll use those, or we'll use a plate form of the same thing. And many laboratories now use molecular tests, either specific DNA extraction and sequencing, or primers specific to particular pathogens. So there's a whole gamut of tests that can be done by diagnostic laboratories. And when you get that report back, with what was found, then you use the synthesis of all this evidence, what you've seen in the greenhouse, and the reports that you get back from the lab, and all of that makes the diagnosis. That's how you determine what's the primary problem, what might be contributing factors, and what is secondary and really not just coincidental and not part of the issue at all. And many times we are ruling out infectious disease. The lab will send back a report that says we couldn't find any primary plant pathogens. That's still very useful, because that's information that allows you not to be thinking along the lines of a pesticide treatment or a biocontrol when there isn't a particular pest pathogen there. So you're saving money in the long run, and can look at more abiotic issues or non-infectious issues to adjust growing conditions to help the plants recover. So ruling out infectious disease is as important as confirming it. And with that, that's the end of my presentation. So Sarah, if there are questions, you can moderate, jump on in now and moderate. And
yeah, thank you so much, Karen. That was I learned a lot myself. Just a reminder to everyone, you can use that same question box that you used to answer the multiple choice questions to type in some questions now if you've got them, and don't worry, I'll ask them anonymously. But Karen, I have a few questions for you just in the meantime while we're waiting for people to jump on, if you've got a minute. I am curious about, you mentioned you use both symptomatic tissue of plants as well as DNA diagnostics when you're sort of figuring out a plant mystery. Can you explain a little bit more about the role of DNA in diagnostics, sort of the interpretation surrounding a report that a grower might get, and also any benefits or limitations to this type of diagnostics?
Sure. I have to say from the outset that I'm sort of an old school diagnostician. So I learned diagnostics before there was a lot of molecular technology out there. But I have tried to keep up with the times. And if I can identify something under the microscope, for me, that's the fastest way. But there are many labs that can do a screen, and they will give you sort of a report that talks about the amount of pathogen that is found. And in my opinion, it's important information, but it's more of a detection than a diagnosis, because there are many groups of fungi. Fusarium is a good example. There are many species of Fusarium that are non-pathogens. They're saprophytes. And then there are some that are definitely primary pathogens and cause plant disease. And so it's important that whatever molecular tests are used are able to discern the difference. And sometimes just giving a genus ID may not provide that information. So the DNA work, some molecular tests are specific for a particular pathogen. And then it's a yes or no, that pathogen is there. Other, it's more of a general information. And if there's a high level of DNA of a particular type, it might make you consider that it's a biotic factor. But you do have to remember that there are Pythium species that are secondary, there are Fusarium species that are secondary and not primary. And so you have to take that information in its context, and I guess. Did I explain that to how you understand it, Sarah? Feel free to
Yeah.
chip in with your experience as well.
Absolutely. And if you have any comments on whether DNA detects both live and dead tissue and how that can be interpreted, or why that's important to put spray records in your form as well.
DNA does, the DNA extraction can, and the molecular methods can detect dead and living pathogens. And so it becomes difficult to interpret, in my opinion. You have to take into consideration what's already been done, because if you've done an application that would have reduced the amount of inoculum of a particular pathogen, you may still detect it with the DNA tests, because it's detecting the dead DNA. So, whether it's difficult to interpret just from a single molecular reading what might really be going on in the crop.
Absolutely, yeah. I would also just add that, sometimes there's secondary infections that come in afterwards, and so just looking at the amount of the different organisms that show up doesn't necessarily mean the highest one's the causative one either. So
that goes back to the quality of the sample as well. If the sample, that also impacts the amount of secondary microorganisms that have gotten a foothold into it if the sample is in poor condition. So you may get a misleading DNA analysis with a poor sample in that regard, and make a misleading diagnosis.
Mm-hmm. I was also curious, just with your 30 years of experience in this now, do you get a sense that in this industry sometimes we underestimate the impact of the abiotic factors that you showed? Maybe because it's nice to think, we can go in and spray something and fix things.
Yeah, we all want to be able to fix things, but so much in disease management, especially in the greenhouse environment, is sanitation and starting clean and growing a healthy plant that can fight back, if you will, and resist and be less susceptible to a lot of microorganisms. And so, having a healthy plant from the outset really is a great way to minimize disease problems. I'd say that, and growing the plant optimally so that nutrition and light and environmental humidity, moisture, stress, all that stuff is minimized. Growing a healthy plant, and then staying clean and starting clean and keeping clean. Sanitation in the greenhouse is another really, really important disease management practice. And if you don't do those two things, spraying a pesticide is really not going to help you a whole lot.
Absolutely. I also wanted to go back to that picture that you asked a question to the group about whether it was abiotic damage or an infection. And it is interesting that people seem to be really split on that one. And that's something I see in the field too, where I think it's, people have a good hold on viruses and fungi, but I think where they get confused is things like bacterial diseases versus some abiotic conditions that can cause that same sort of leaf spot or necrosis. Other than checking the underside of the leaf, do you have any other sort of tips or tricks to distinguishing abiotic factors from sort of those bacterial leaf spots that may mimic that?
Well, again, we go back to the patterns, and it was sort of unfair of me to show an individual leaf and not show you a whole bench full of those plants, because if you could see the bench, all the plants would show spotting, and it would all show up within a couple of days, sometimes even one day of an application, as opposed to a bacterial leaf spot disease, which will start in one area or maybe a few areas and then kind of spread through the plant, as well as spread plant to plant over time. And by time, it can be weeks, 2 weeks, 3 weeks sometimes. And that would be a real clue to help you as well, to determine that, if everything's spotted up within a day or two of an application, odds are that's a non-infectious issue and probably related to some sort of phytotoxic response.
Okay, someone online has a question that I was hoping to ask too. They were wondering about if you have a story that's sort of one of your most interesting or challenging diagnostic stories, something that you were surprised by or came out to be the opposite of what you expected it to be.
Wow. Well, diagnosticians always have a cadre of stories to tell. I'm trying to think of some that were, well, a lot of times, that to be honest, that one that I showed you was a bit of a surprise to me as well, because I spent a lot of time cutting into those leaves looking for bacterial streaming and incubated them looking for fungi. But when I realized you turn them over, there wasn't any kind of damage on the underside. I've also seen issues. One that was really interesting was poinsettias have a very strange response to chlorine. And if anybody had used chlorine as a disinfectant, even on greenhouse benches and things, if there was some sort of slight absorption on poinsettias of the roots, they get these very bizarre black target-shaped lesions on the stems. And at first, I thought that this was a viral issue. I mean, it kind of looked like impatient necrotic spot virus on a poinsettia, but we were able to duplicate it with exposure to something like a 10% sodium hypochlorite solution. And when it was absorbed through the roots, you got these very bizarre target shapes. It was like bullseyes on the stems of poinsettias in black. So that was one that was very odd, and took a lot of trial and error to figure out. I'm always surprised by the pathogens and different cultivars will respond differently to a particular disease as well. There may be more or less yellowing associated with a leaf spot. And so that's always a real challenge, is that, the new cultivars are constantly being developed. And sometimes the diseases present quite differently. Even though the species of plant is the same, it presents differently with a cultivar that's more resistant or susceptible.
Yeah, I've noticed that myself, even with viruses, where you'll have a greenhouse and one cultivar won't show any symptoms at all, and another one's completely hit by viral symptoms. And it is challenging to know sometimes whether that's part of the cultivar or, something else.
Yes, absolutely. Impatient necrotic spot is really the master of disguise. I mean, it produces very bizarre symptoms on many different kinds of plants. And they don't look like each other. Dark stem lesions or bleached stem lesions, and sometimes it's mottling and sometimes it's ring spot, but it's all the same virus. It's really quite remarkable what the pathogens can do.
And do you have a sort of sense for growers whether foliar diseases are easier for them to sort of diagnose on farm, or root rot should maybe always be sent off for diagnosis, or what's your feeling on that?
I feel like growers probably should get a diagnosis of root rot diseases, at least initially, particularly if it's a new crop that they're growing. You can learn from those diagnostic experiences and maybe assume that next season you will see a similar issue with that particular plant. So you don't necessarily have to send it out every year. But the symptoms can be deceiving, and I think it's important to know what root rot pathogens you might be struggling with, and then try and figure out where the source of those are, whether it be the water source or maybe a plant source. A lot of things can come in with plants that are purchased. So I think it's important to get a lab, a confirmation of a diagnosis, certainly for anything that's really strange. I don't know that, leaf spot versus root rot, if there's really a difference, because experienced growers will recognize symptoms of both over time, and particularly when it's a particular host. They're familiar with the diseases that they get. But so you learn from those diagnoses. So I think it's important to get a confirmed diagnosis either way. And that helps you. I mean, things like powdery mildew, I think most people will recognize. And so maybe that's not something that, or even downy mildews will not necessarily need to be sent out to a lab. But you should always consider sending it to a lab if you're not sure, rather than guess. It's more important to test than to just guess.
So we've got two more quick questions, and then we'll hand it over to Greta. Someone was asking, are there telltale signs of pathogenic fusarium? To know for sure it's fusarium, do you see like a red-orange color on the roots and a stem canker or wilting, or is there a way to distinguish fusarium, or do you have to send it off to the lab?
Root rots are particularly different. In some cases, there's a reddish discoloration with fusarium. Not every single time. But if you know the host crop and you know it's susceptible to a fusarium crown and root rot, maybe something like Lisianthus, or if it's a vascular wilt and you'll see the discoloration in the vascular system, that would be clues that you might be dealing with a fusarium. But I have seen fusarium root rot issues that aren't, and the roots are not red; they're just brown and decaying, and they've got lesions or they're falling apart. So it can be tricky. Just symptoms are the plant's response to a pathogen, and they have a kind of a limited repertoire of response, I guess. So dark roots can be caused by a lot of things.
And lastly, someone had a question about some of the fantastic pictures you showed in your presentation. A lot of them were referenced as bugwood.org. I think a lot of us maybe up in Canada are unfamiliar with that. So what kind of website is that? Is that just a catalog of pictures of diseases? Is it useful for growers to search through?
Bugwood is started out, I believe, at the University of Georgia and is still there, but it is a website where a lot of plant pathologists and researchers in the States have deposited their images. And you can go there. If you just Google Bugwood, you'll see several different image collections, and you can become a member. And these are images for use for educational purposes, such as this webinar today. And they tell you how to credit them, and that's why you saw the credits on those pictures. And I find it really interesting. I like to go, people all over the country have deposited images there, and so there are things there that I have not seen. It's not necessarily cataloged as easily for a learning experience, but if you can search based on the pathogen or on the crop, and you can see what comes up, and they're all labeled. So in that way, you can kind of peruse and see if you see something that might look similar to what you are seeing. You may have to sort through 100 or more images, but it is a good resource, an excellent resource for education.
Excellent. Well, thank you so much, Karen. And I'm just going to pass it off to Greta to wrap us up.
Oh, thank you. Thank you for the wonderful presentation, Karen, and Sarah for the great moderation and discussion today. As a reminder to the audience, a copy of the webinar recording will be sent out tomorrow to everyone who's registered and is available online. For more great content on greenhouse ornamental production, visit onfloriculture.wordpress.com, which is run by Sarah and her colleagues at OMAFRA. And of course, check out greenhousecanada.com for news and production tips. Be sure to register for our 4th webinar in the series happening next Tuesday, April 9th. We'll be looking at ways to deal with powdery mildew with Dr. Jean Williams Woodward from the University of Georgia. See you next time.